In this lecture, we're going to talk about the processes of mitosis and meiosis, essentially the cellular division processes, which are either creating two copies of the maternal cell or lead to the formation of gametes and enable sexual reproduction. Before we get to the whole conversation about mitosis and cell cycle, we need to remind ourselves about the organization of cellular genome. In prokaryotes, genome consists of the single circular chromosome, essentially one circular double-stranded DNA molecule within the nucleoid region, a special part of the cytoplasm where the DNA is. In some prokaryotes, you can find also plasmids, small circular DNA that are not essential for microbial growth, but can carry certain beneficial genes. Plasmids can be exchanged between bacterial cells, and the genes that bacteria will receive may or may not be added to the chromosome, but they will essentially confer some new traits to the bacterial cell that received the plasmid. Unlike prokaryotes, eukaryotes have more than one chromosome, and these chromosomes are linear. Each of these linear chromosomes is a double-stranded DNA molecule as well. The number of chromosomes in the nuclei of the somatic cells is different between different species. For instance, in humans, diploid cells that are, most of them are somatic cells, have 46 chromosomes. On the other hand, the human gametes, which are haploid cells, have 23 chromosomes. Why do somatic cells have double the number of chromosomes compared to gametes? A somatic or diploid cell contains two homologous sets of chromosomes, with one set coming from each biological parent. 23 chromosomes from a mother and 23 chromosomes from a father, which makes the total number of chromosomes 46. In gametes, on the other hand, gametes such as sperm cells or oocytes, we have a haploid set, one set of chromosomes, 23. During fertilization, when spermatozoa fuse with secondary oocyte, the resulting cell, the zygote, receives one set of chromosomes from each gamete. In other words, a zygote becomes a diploid cell with 23 homologous pairs. Homologous chromosomes have the same length, and they have the same genes that can be found in the same locations, or loci. Locus is the singular form, and these genes encode specific proteins. These proteins, in turn, determine the traits. So, as we mentioned before, in a homologous pair, each of these chromosomes is from a different parent, which means these two chromosomes will carry different alleles of a gene for the same trait. Alleles are not identical. And due to the combination of the alleles, humans have phenotypic variation. Let's take a look at the example of the ABO blood group. In the blood group ABO, there are three possible alleles for the blood type, A, B, or 0, which essentially encode certain carbohydrates on the surface of the red blood cells. All human cells have two copies of this allele. So the blood type, in this situation it is the trait, is determined by two inherited alleles of the gene of interest in a pair. So what combinations can be? You can have same genes, same alleles on the both chromosomes, AA, BB, or OO, or two different genes, AB, AO, or BO. As a result, if the combination is AA or AO, the blood type is A. BB or BO, the blood type is B. Combination BA makes AB blood type, and combination OO forms the O blood type. There is one exception to the total similarity between homologous chromosomes. It's sex chromosomes. So there's a homologous pair, 23rd pair, that consists of two sex chromosomes. If sex chromosomes are X and Y, it's a male. If sex chromosomes are X and X, it's a female. As you can imagine, genes that are present on the sex chromosome X, that is inherited from one parent, and sex chromosome Y, which is inherited from another parent, these genes, these alleles, are not identical because chromosomes are not identical. DNA is enormously long. If we would stretch a molecule of DNA from a human cell, it would have a length of about 2 meters with a diameter of only 2 nanometers. However, the size of a cell is roughly 10 micrometers. So fitting 2 meters of DNA into 10 micrometer cell requires a very, very tight package. That packaging should also provide a compromise to make DNA accessible for gene expression. This is why DNA in chromosomes is compacted. The first level of compaction is when short parts of DNA wrap around histone proteins. Here you can see a bunch of histones and the DNA wrapped around it. These 8 histone proteins and base pairs of DNA form what's called a nucleosome, the histone DNA complex. Nucleosomes are located at equal intervals along the chromosome, and nucleosomes are connected by so-called linker DNA. At the second level of compaction, nucleosomes and linker DNA fold into a 30 nanometer fiber. And finally, the third level of compaction is when fibrous proteins pack the chromatin. So this packaging ensures that in a non-dividing cell, each chromosome occupies a particular area within the nucleus. DNA only replicates in one of the phases of the cell cycle called the S phase. S phase is not the part of mitosis, cellular division, but it must precede mitosis in order to provide a DNA copy sufficient for 2 daughter cells. When a chromosome is replicated, it is composed of 2 linked sister chromatids. This illustration of the metaphase chromosome right here shows you 2 sister chromatids that are bound to each other by cohesin proteins located at the center of the chromosome. That connection is closest at the centromere. So this area is called a centromere, and these 2 are sister chromatids. So now we can talk about the cell cycle, which is a highly ordered series of cellular events in which cell grows, replicates its DNA, and divides into two daughter cells. Cell cycle is exquisitely timed and tightly regulated. It consists of two major phases, interphase and mitosis. During interphase, cell grows in size, its DNA is replicated, and it generally gets ready for mitosis. During mitosis, also known as karyokinesis, the replicated DNA is separated. Karyokinesis can be translated as the nuclear division. Once the nucleus is divided, cytoplasm is divided as well by the process called cytokinesis. Interestingly enough, in some cells, cytokinesis may not happen. In this case, if cytoplasm is not divided, the multinucleated cells are going to be produced. So the first phase of the cell cycle is the interphase. During interphase, cells grow and prepare for cell division. Interphase consists of three specific stages: G1, S, and G2. During G1 phase, also known as the first gap phase, the building blocks that are essential for the chromosomal DNA synthesis and for the synthesis of proteins are accumulated, and cell reserves sufficient energy to complete the replication of the chromosomes, because the process of DNA synthesis is anabolic and endergonic. In other words, it requires energy. During S phase, cellular DNA is replicated, forming sister chromatids that, as we talked before, are attached to each other in the centromeric region. Centrosomes, structures that participate in the process of cell division, are duplicated, and centrosomes will become mitotic spindle that will move the chromosomes. In the second gap phase, known as G2, cell replenishes its energy storage to deliver enough energy for the process of mitosis. Proteins that are essential for the separation of the chromosomes are synthesized, organelles are duplicated, and the cytoskeleton is degraded. Once the interphase is complete, mitosis starts. This is the multi-step process in which duplicated chromosomes, each consisting of two sister chromatids, are aligned at the equator of the cell. Sister chromatids are separated and moved into two identical daughter cells. The first portion of mitosis is called karyokinesis, or nuclear division. The second portion is cytokinesis, which implies physical separation of this nuclei and formation of two daughter cells. Karyokinesis, or mitosis, is divided into prophase, prometaphase, metaphase, anaphase, and telophase. So what is happening during prophase? Since chromosomes have to be separated into two cells, they cannot longer stay in the nucleus, so the nuclear envelope starts to dissociate. Membranous organelles undergo fragmentation and move towards the periphery of the cell, and nucleolus disappears. Centrosomes begin to move to the opposite poles of the cell, extending microtubules from one centrosome to another, pushing centrosomes farther apart as microtubules increase in length, which makes the cell a little longer. Sister chromatids at this point start to coil with the participation of condensing proteins, which facilitate that coiling, and now can be seen under the light microscope. The next phase, pro-metaphase, is characterized first of all by the complete elimination of the nuclear envelope. Mitotic spindle, as you can see, keeps developing, since more microtubules stretch across the area of the former nucleus. Chromosomes become more condensed, more discrete, more visible, and each sister chromatid develops a special protein structure called a kinetochore in the centromeric region. Here you can see a kinetochore. Proteins that comprise kinetochore bind to the microtubules of the mitotic spindle. The fiber of microtubules touches to chromosome, it orients the chromosome, and eventually sister chromatids face the opposite poles of the cells. Well, not specifically chromatids, but kinetochores of two sister chromatids. You can see it in this illustration that these kinetochores are facing the opposite poles of the cell. Now, there is another kind of microtubule, so-called polar microtubules. These are not bound to kinetochores on the chromosomes. They overlap between two poles, here and here, and contribute to further cell elongation. Now, here we can see a better rendition of the centromeric region of a chromosome and kinetochores that are attached to two sister chromatids as well as to mitotic spindle microtubules. During metaphase, chromosomes align in the equatorial plane between two cell poles, between two centrosomes. Remember that sister chromatids are attached to each other by cohesin proteins, and chromosomes at this point are condensed. But during anaphase, cohesins that hold two sister chromatids at the centromere degrade. So sister chromatids can separate. At this point, each chromatid earns the status of a single chromosome, and it is pulled towards the centrosome, towards the cell pole, by its specific microtubule. At this point, polar microtubules slide against each other at the metaphase plate, elongating the cell. And once sister chromatids are fully separated and turned into assembly of the chromosomes, telophase starts. So in telophase, you can see that chromosomes are at the opposite poles of the cell, and they start to decondense into chromatin. Microtubules that form mitotic spindle disassemble, and nuclear envelope starts to form around the chromosomes. Nucleosomes start to appear. There is a special phase for some cells that are not actively dividing, called G0. so-called quiescent or inactive stage. It may be temporary or permanent. For instance, it can be temporary if nutrients or growth factors are not available, and cell will stay in G0 or quiescent phase until the conditions will be suitable for division, or some external signals will trigger the start of first gap phase. However, there are cells that never divide. For example, red blood cells, neurons, or cardiomyocytes. These cells remain in quiescent phase permanently. Now I want to go back to telophase and remind you that there's a process called cytokinesis here. So actin proteins, they form the cleavage furrow, contract, and separate two cells. Now, we mentioned that cell cycle is very well regulated. Now, cell division can be triggered by a variety of external stimuli, such as death of the cells nearby, hormonal release. For instance, human growth hormone results in the stimulation of cell division, and its absence inhibits cell division, resulting in dwarfism. In excess, human growth hormone leads to gigantism. Another type of external stimulus that can lead to inhibition of cell division is contact inhibition, when cells become very, very tight and touching each other. When cell grows, its surface to volume ratio decreases, which, in order to facilitate the survival, promotes cell division. Now, it's important to understand that if the process of DNA synthesis during chromosomal duplication or distribution of the chromosomes during the anaphase, they carry mistakes, it may result in mutations in daughter cells. And these mutations may actually be passed down to next and next generations of cells. To ensure that the cell cycle operates smoothly, there are so-called cell cycle checkpoints. They regulate the progression of a cell to the next stage and can stop it if needed. The very first checkpoint is G1 checkpoint, or so-called restriction point. It is at the end of G1, and it checks for availability of reserves, energy, size of the cell, and the DNA damage before it is replicated. If the cell passes the checkpoint, it irreversibly commits to mitosis. Growth factors actively participate in passing the G1 checkpoint. If the cell doesn't meet the requirements, either there are not enough available resources, or DNA is damaged, cell does not progress to S phase and attempts to fix the problem, or go into quiescent G0 stage. So we're going to kick it. Accidental copying. At G2 checkpoint prevents the cell from entering mitosis by assessing the size of the cell, the protein reserves, and by ensuring that all chromosomes are correctly replicated and DNA isn't damaged. If DNA is damaged after the replication, the cell cycle stops again. Cell may attempt to complete DNA replication or repair the damage. And if that happens and it passes the checkpoint, the last checkpoint is M checkpoint. It is at the end of the metaphase, so-called spindle checkpoint. The question that cell asks itself, sort of, whether or not sister chromatids are correctly attached to the spindle. Because when chromatids are separated in anaphase, this process is irreversible. Which means if they are incorrectly attached, one daughter cell can have more chromosomes than it should, and another one may have less. And at M checkpoint, the cycle wouldn't proceed if each pair of sister chromatids is anchored to at least 2 spindle fibers that arise from opposite cell poles, and that's really important. So cell cycle can be regulated positively or negatively. In positive regulation, intracellular molecules can promote cell division. Well, in negative, they can stop. So the molecules that regulate cell cycle may act by themselves, or they may regulate other proteins, other enzymes, other factors. And the best known example of positive regulators of the cell cycle are cyclins and cyclin-dependent kinases, or CDKs. They regulate the progression of the cell through the checkpoints. The levels of cyclins change throughout the cell cycle, as you can see from that graph. The levels of these proteins are regulated by external or internal signals. Interestingly enough, when cell moves to the next stage of the cyclin, look at cyclin E, which is active between G1 and S phase. When cell moves to G2 phase, cyclin E is gone. So cyclins that were active previously were degraded. Now, in order to regulate cell cycle, cyclins must bind to cyclin-dependent kinases, CDKs. These complexes between CDK and cyclin can be phosphorylated, and that phosphorylation activates them. You can see here on that panel that cyclin binds to CDK, then the enzyme called kinase phosphorylates CDK cyclin complex. So now CDK cyclin is activated. Phosphorylation activates the protein because addition of the negatively charged phosphate group changes its shape. Well, CDKs are also kinases, so they also can phosphorylate, but other proteins. So the proteins that CDK will phosphorylate are ultimately critical for advancing the cell cycle. Here you can see how CDK cyclin complex transfers phosphate group onto a target protein. Now, the levels of CDK are relatively stable at each phase of the cell cycle, but it's the cyclin concentrations that will determine when and whether CDK cyclin, in which CDK cyclin complexes form. It's conceivable that different cyclins and CDKs will form complexes at different points in the cell cycle to regulate different checkpoints. Now, cyclin levels are based on the timing of the cell cycle, which means that its regulation occurs either by CDK alone or by CDK cyclin complexes. If there are no cyclin CDK complexes, no checkpoint progression. So-called CDK inhibitors may prevent the activation of CDK and monitor a particular cell cycle event. Now, the most famous negative regulators of the cell cycle, which stop it, are tumor suppressor proteins, such as retinoblastoma protein RB, P53, and P21. These proteins act at the G1 checkpoint, and they are called tumor suppressor proteins because when the genes for these proteins are damaged or non-functional, cells would uncontrollably replicate. P53 is a multifunctional protein that responds to DNA damage. If DNA damage is detected, P53 stops the cell cycle and initiates the repair of the DNA. If DNA cannot be repaired, P53 sends the cell into apoptosis, programmed or controlled cell death. If cell dies, damaged DNA is not copied and cannot be passed to the next generation of cells. Now, how does p53 stops the cycle? When p53 is activated, it in turn activates p21 enzyme. p21 binds to CDK cyclin complexes and inhibit the activity. And you may remember that active CDK cyclin complexes are essential for passing through the checkpoint. So, for instance, when cell is under the stress, the levels of p53 and p21 increase, which means cell is probably not going to move into the S phase. Another tumor suppressor protein, retinoblastoma protein, named this way because this gene is mutated in retinoblastoma, the cancer cells, monitors the cell size. When retinoblastoma protein is dephosphorylated, it binds to transcription factors, for instance, E2F. When retinoblastoma protein is bound to E2F, it inhibits it. Therefore, this factor cannot bind the DNA, and the proteins that are essential for transitioning from G1 to S, their production is blocked. When cell size increases, the retinoblastoma protein is phosphorylated. Phosphorylation inactivates it, which means it doesn't bind E2F transcription factor anymore. The block on the transition from G1 to S is removed. So in brief, for the cell to move past checkpoints, the positive regulators of the cycle must be on, while the negative regulators must be off. Now, we mentioned that sometimes cells go into uncontrolled cell growth. We call it cancer. So, for instance, DNA accumulates some mutations as a result of replication errors, and a small number of these mutations will be passed to the daughter cell. If this mutation is in the gene for the protein that controls the cell cycle, the protein will become faulty, which will produce errors in the control of cell cycle. So if mutations that affect cell cycle accumulate, that decreases the effectiveness of the cell cycle control and repair, which means cellular division will speed up. Proto-oncogenes, in this case, serve as positive regulators. Now, we must remember, proto-oncogenes are positive regulators that facilitate cell division, and they are normal. However, when proto-oncogenes accumulate mutations, they can turn into the oncogenes, which are altered proteins, and the proteins will increase the rate of the cell division. Now, normally, the mutation in proto-oncogene will result in the faulty protein, and the cell cycle is not completed. But in case of cancer, mutation does not inactivate the protein. It actually increases its activity as a positive regulator. For instance, what if CDK is activated independently of cyclin? In this case, cell cycle will pass the checkpoint even if the conditions that are required to clear the checkpoint are not met. If this daughter cell that is now atypical can divide as well, more mutations accumulate. Before we move on, I want to highlight something, that cells have a lifespan. They live, some of them die, but basically all the population of cells in the stationary state. The tumor may result from increased cell division when the rate of cell death is normal, or from the normal cell division, but when apoptosis, for instance, the one triggered by p53, is decreased. So let's talk a little bit more about tumor suppressor genes. As we mentioned before, they code for negative regulators. So these proteins, retinoblastoma p53 and p21, they stop cell cycle progression until the conditions that are required to move to the next phase are met. If the negative regulator is mutated, cell cycle cannot stop. Which means mutated p53 is probably found in many cancers, and it is true indeed. When p53 is mutated, the protein is faulty, and DNA errors are not repaired or detected. Functional p53 will trigger apoptosis in case of DNA errors. Damaged p53 cannot do that, and while cells divide fairly normally, they don't die, and they are faulty cells. Also, when p53 is dysfunctional, the production of protein p21 practically ceases, which means cyclin-dependent kinases are not activated and the G1 checkpoint is compromised. Cyclin-dependent kinases are constantly activated, which means if CDKs are active, p53 is dysfunctional, and p21 is dysfunctional, cell can move from G1 to S phase regardless of whether it is ready for it or not, which means daughter cell will inherit the mutations in p53 gene. And further accumulation of both oncogenes and non-functional tumor suppressor genes is absolutely conceivable if you think about the fact that the cells divide very rapidly. So here you can see that in case of DNA damage, or stress, or hypoxia, p53 is supposed to lead to cell cycle arrest. If p53 is faulty, it doesn't, and cell cycle continues. If cell is abnormal, p53 is functional, it will either end in the restart of the cell cycle or programmed cell death. But if it is dysfunctional, the accumulation of further mutations may lead to the cell becoming cancerous. Now, the process of mitosis is characteristic for eukaryotes. In prokaryotes, cells divide by so-called binary fission. It is less complicated and more rapid for a variety of reasons, mostly because there's only one chromosome, it's relatively small, and no histones are involved. So, the chromosome in a prokaryote is circular, and it's in a nucleoid, so there's no complication in the form of nuclear envelope. So no histones and nucleosomes, which means less work on unwrapping that DNA. Now, chromosomes in prokaryotes are attached to plasma membrane, and close to attachment site is the origin of replication, which, as you may remember from previous conversations, is bidirectional, which means replication forks move away from the origin of replication on both strands. It results in the formation of two new double strands, and the origins, marked red on this image, move towards the opposite poles of the cell. Once the chromosomes clear the midpoint of the cell starts the separation of the cytoplasm. FTSZ protein, shown here in green beads, which stands for filamenting temperature-sensitive mutant, FTSZ protein forms a ring and partitions nucleoids. Eventually, the septum is formed between the daughter cells, new cell walls form, and the cells separate. Now, this is the first part of the lecture video. Second part will be dedicated to meiosis.